It's a real treat to be part of such an eclectic gathering. As Tim said, this will be slightly different, actually quite different from what has gone before. One of the things that has impressed me a lot about the earlier talks is how sort of directed the search was. And that's not the case here for the little example I want to relate. This actually didn't begin as a drug discovery project. It may well not end as a drug discovery project, but here's where it started. So it started in a project on ants. That's an ant nest in Brazil. A team of Brazilian and German researchers came and pumped about 10 tons of concrete into the ground, let it sit for a while, and then started excavating. And what you can see is one very large ant nest. It probably had 5 to 10 million ants in it before the concrete arrived. It had probably existed for close to a decade. And it is one of the most highly evolved and charismatic of a whole group of ants, about 200 species, all in New World tropics, neotropics, that have evolved to grow fungi. And they go out, they're the leafcutter ants, and many of them, and they go out, they collect plant material, they carry it back to the nest, they carry it below the ground, and in their nest, they have these gardens, these sort of football-shaped objects, and in there's a fungus, and they feed the plants to the fungus, and then they eat the fungus. So that's why they're called fungus-growing ants. And that system has been known since the late 1800s in some detail. Starting about 15 years ago, there were a couple of discoveries that changed our view of that, and that's why we became interested in them. And the two discoveries are shown here. I don't know how well you can see it, but there are two ants, and they're down in their fungal garden, and you can see that the ants themselves are covered with some white stuff. Here's still another of the fungus-growing ants in its garden, and you can see that there are these black specks in the garden, and that turns out to be another kind of fungus that's growing in the garden. And those two discoveries are in fact related. The white stuff on the ants, which was thought to be insect wax, turned out to be bacteria. The ants carry a bacteria that they feed, and it's a highly symbiotic association. The black thing turns out to be a different kind of fungus. The food fungus is like a mushroom. The black thing is like, well, what you see in your shower. They're two very different kinds of fungus, and the black one is a fungal pathogen. And it can wipe out the nest, and can wipe out the whole colony of ants. And then the relation between the bacteria and the pathogen is that the bacteria actually make an antifungal agent, doesn't kill the crop fungus, does kill the pathogenic fungus, doesn't kill the ant. And so it's a very simple symbiotic system. You don't find the ant without the fungus, you don't find the fungus without the ant, and so they benefit each other. But with the addition of the pathogen and the bacteria, it became a more complicated system. So the bacteria suppress the pathogen. The pathogen would have suppressed that, so on the theory, and that helps that, so on the theory that the enemy of my enemy is my friend, the bacteria help the ant, and the ant feeds the bacteria, right? So it's a very complicated but interesting system. And what we were interested in was this step right here. That is, how do the bacteria suppress the fungus around some other things that it doesn't want to suppress. And so it was that selective suppression that seemed interesting, and bacteria always do that with small molecules. And so we thought that would be a way to discover small molecules and learn something about the chemical ecology of this multilateral symbiosis. And so starting then, now what seems like a long time ago, we started to work on one of the ants, shown here, and we found and characterized the antifungal agent that it produces, and that's shown here. And I won't say anything in detail about these structures. If they either are the reason you love organic chemistry and natural products, or the reason why you became a clinician. I mean, it's complicated and has a number of interesting features. So, so the question is, all right, you've done that once. What do you do next? And our answer was, you do it several more times, because you could learn something new each time, because this changes and the environment changes. And so it's sort of an algorithmic way to keep finding interesting small molecules. So I'll talk about two examples of those kinds of discoveries here. One is that it can reveal things that are called cryptic metabolites, and the other, probably more interesting to the audience, is that it suggests a way to find therapeutic agents. Now, what's a cryptic metabolite? What I'm showing here is sort of a representation of the genome of the bacteria that makes erythromycin. Okay? Erythromycin has been known essentially forever. It was discovered way back in 1949. It became used in humans in 1952. That's the way things were in those days. And erythromycin we grow on ton quantities. And it really is a bacteria that we know quite well. And so we certainly know the genes, right, sort of symbolically represented here, the genes that encode the enzymes that give us erythromycin. And that was all we knew until about 10 years ago, when the whole genome of the erythromycin producer was made. And what it revealed was that there were actually a lot of these clusters of genes that could make things that were like erythromycin. There were clusters of genes that could make things that were like vancomycin. And there were genes that could make hybrid molecules using the biosynthetic strategy for both of those. And so that was 10 years ago. And at that time, if you asked how many of these ones in red, how many of those did we know, what the molecule was, what it might do, the answer was very simple. It was 0. Okay? And you say, okay, you've had 10 years to work on it. How many do you know? Same answer, 0. Okay? So that's the issue, right? It's an opportunity to make a lot of discoveries, things that could be as useful as erythromycin, maybe even more useful, but it's also clearly a tough technical problem. And our sort of epic failure to find these molecules was solved in the typical way these kinds of things are. They were just named cryptic metabolites. How could we be expected to find them? They were hiding. They were cryptic. And so one of the features of this symbiotic system is that it offers an alternative, it offers a way to uncover some of these cryptic metabolites. And so this happens to be the genome of one of the ants. It's a molecule like the molecule I showed you at the beginning, but if you look through, and that molecule comes from some genes here and some genes here on a plasmid, But if you look all through the genome, you find all of these clusters, right? So it's not just the erythromycin producer. It's literally every bacteria that we've ever grown has the ability to produce maybe an order of magnitude more things that we don't see. So how to go about dealing with that? A number of labs are working on a number of ways, but one way is to ask, why are cryptic metabolites cryptic? And they're cryptic because they're actually only produced on an as-needed basis, and we don't understand what that as-needed basis is. That is, there's something out in the environment that produces a signal that the bacteria responds to by making small molecules. So bacteria sense and deal with the world largely through small molecules. So based on that, you can then begin setting up things that would stimulate the production of these cryptic metabolites. And I'll show you one way we went about addressing that issue. But before I can do that, I have to tell you a little bit more about how the ant system is organized. So this is a relatively recent paper, and what it shows is it traces nitrogen sort of through the nest, and so you can figure out what the food chain is, what the trophic levels are. And so plants are nitrogen poor, and so about half the nitrogen that reaches the crop fungus comes from plants. The other half comes from nitrogen-fixing bacteria that are in the nest. And then you can then see through this stable isotope labeling that there's 1 degree of fractionation, both for the ants, because the ants eat the crop, and for the pathogen, so the pathogen also destroys the crop. It's not a weed, it's really a pathogen. And then at the top of the food chain actually is a bacteria. So the bacteria is fed by the ants. So we call it the ant system, but we could also call it the bacterial system because it's at the top. Now, if you then ask, what are the greatest existential threats to the bacteria? One, we already know. That is, it would be the pathogenic fungus for reasons that we've gone through. But if you think about it, you realize that there's another, actually, existential threat to the bacteria, and that is a bacteria that's exactly like itself, okay? That is, I mean, it's the same way in scientific research. Your greatest competitors are the people who are most like you, right? And so among all these ants then, all these species of ants and all their species of bacteria, what these bacteria have to fear is another bacteria that could come in and displace them. And so we set up then, so it's like a bacterial Game of Thrones, right? It's all these crazy things whose names you can't remember, who are fighting with each other and forming these complex alliances and enmities. So, the assay we used in this little example is you could take one of the ant bacteria, put it on a petri dish, let it grow for about 2 weeks, then takes the bacteria from some other ant, spread it around the outside, let it grow for 2 weeks, and then see whether there was a zone of inhibition or not. That was called, for obvious reasons, the intruder assay. And then you could do a high-tech measurement by taking a ruler and measuring how big the zone of inhibition was, and then you could glitz it up for a publication by making it into a heat map. Okay? So that was the strategy we used for discovery. One of the things you can see from this is that there are 2 bacteria, one called BCI1, one called BCI2, that couldn't be more different, right? BCI1, complete wimp, can't prevent intrusion by any of the other bacteria. BCI2 is pretty studly. It can resist everything except itself, okay? That was very surprising. That's not at all the result we expected, because in fact, we had every reason to believe that BCI1 and BCI2 were exactly the same. And the reason was that BCI stands for Barrow, Colorado Island. It's a small island in the Panama Canal. either in the entrance or the exit, depending on which way you're going. And it's a national park in Panama. It's operated by the Smithsonian Tropical Research Institute. And the two nests were nearby on this island. If you ask the entomologists about the ants, they would say, yeah, they're the same. Taxonomically, the ants were the same. And actually, if you looked at the genomes of the two ants, they were virtually identical. Yet they behaved completely differently in this phenotypic assay. That was what was surprising. It turned out that the reason they were different is they could make this molecule here, which is a relative of actually a very interesting molecule, which ironically we had discovered in different circumstances many years earlier, called rebecamycin, which is a relative in turn of staurosporin, which has sort of a storied history as starting the selective kinase inhibitors. And so this turns out to be, among other things, an extraordinarily active antibacterial agent. But how could two bacteria with essentially identical genomes have such different chemistry? And the reason for that is shown here, that while the two genomes were essentially identical, and they had two linear plasmids, which were virtually identical, the genes just came in a different order. One of them carried a circular plasmid, while the other carried a linear plasmid. And that circular plasmid is the one that happened to make the potent antibacterial agent. So it was not sort of, if you will, normal genetic inheritance of the ant. It was on a plasmid. And that actually is quite interesting, okay? So that's the background to then how we set about trying to do some more systematic discovery. And from what I've told you so far, if, let's say, you were preparing an NIH application and someone said, what are you going to actually use this for? The best answer, I think, would be, we're going to use it to discover antifungal agents. And so one slide on why you might want to discover antifungal agents. So this is actually an extraordinarily interesting paper from 2012. And what it looks at is what turns out to be an enormous unmet medical need for invasive or systemic fungal infections. That is, we have ways that are moderately okay to control superficial fungal infections. But once a fungal infection gets inside, we have but few poor ways to control them. And so it's really actually quite surprising when you look. Here are the number of cases. Here's the mortality rate. And just to calibrate you on those mortality rates, for Ebola, it's about 50%. Right? So these have mortality rates that sort of start there and go up. And here are two that I highlighted. One is for an Aspergillus fumigatus. The other one is from Candida. And you can see the reason these have flown under the radar is that the number of cases is relatively small, but the seriousness then is shown by the high mortality rates. And as this article points out, that once you realize that probably more people on a worldwide scale die of these invasive fungal infections than die of TB or malaria. Okay? And while certainly mortality rates for TB and malaria are going down, for fungal infections, they're not going down. In fact, if anything, they're picking up and new fungal diseases are appearing. So that's the case for why one would want to look for antifungal agents. So we went about looking for them in using a strategy you now understand. You would take bacteria from ant nests, You would expose them to pathogens, the pathogenic fungus. That fungus would give off some signal. They would think they were under attack, or their nest was under attack, and so they would produce molecules that would suppress the fungal pathogen. And so that's what we were looking for. And the study I'll tell you about involves 3 ant nests from a location called La Selva. And these 3 nests were all next to each other virtually. They occupied about the area of this lecture room. So one there, one back there, one over here. So there were these 3 nests, and they all seemed to make something that would suppress various fungal pathogens. Now, as it turns out, the molecule that they all made the same molecule, nest 1, nest 2, nest 3. And it's a molecule that I show there, and we gave it the name Selvamicin because the place in Costa Rica where we discovered it is called La Selva. And if you look at it, you say, yeah, it's a molecule. If you look at it and you know a little bit more about antifungal agents, you'd say it looks an awful lot like 2, amphotericin and nystatin. The good news about those are they're both on the WHO list of essential medicines. The bad news is they have terrible liabilities. They have pharmacokinetic liabilities. So, for example, solubility, there is none. They have biological liabilities, probably the most extreme of which is death, which is about as extreme as a liability can be in a medicine. And so while they're used, they're far from perfect. And so you can see this has, in many ways, resembles these 2. But for whatever reason, whether it's because the shorter stretch of these double bonds, or the fact that it doesn't have the zwitterionic end of those, or this extra sugar that's on it, for whatever reason, what we knew sort of immediately is that this had much better solubility properties than these 2. Okay, that's a low bar. But it also had much greater stability than these 2. So we began to investigate that a little more. Now, what was interesting, I think in many ways most interesting about this, was that while they all made the same molecule, for 2 of them, not for 3, but for 1 and 2, we had the complete genome. Okay. Why did we just get the complete genome for 2 things that we thought were identical? It was a screw up, but it turned out to be an interesting screw up. If you look at the biosynthetic genes that are contained in 1 or 2 that make that, they are essentially identical, right? 99% base pair by base pair identical. And you'd say that's hardly surprising. They make the same molecule. They should have the same genes. But what is surprising, and continues to be interesting about this system, is in one case, the genes are integrated into the chromosome, and in the other case, the genes are on the circular plasmid. This just happens to be, Ethan always likes to draw things this way to emphasize that plasmids are much smaller than chromosomes. So that's what's interesting. The chromosomes of the 2 bacteria are only 83% identical, but the plasmid that they share is 99% identical. So it tells you somehow this is like a plasmid-based chemical defense strategy where they're sharing these things. Now, the world doesn't need probably a slightly better version of amphotericin or nystatin. One of the ways, it's not completely known how these work, but it's thought that they work by binding ergosterol that's in the plasma membrane of the fungus. And so a diagnostic test for that mechanism of action is shown here. You make tiny little vesicles, you put ergosterol in, and then you drip in your drug. As you put in a little drip of the drug, heat's released, and then you wait, cool that back, then you add another one, cool it back. So it's this isothermal titration calorimetry. And so that's what that shows, and that's what's true for all of the polyene antifungal agents. We did this with Selvamicin, fully expecting that it would look exactly like that. It doesn't. It's flatline. So its mechanism is completely different from what we would have expected. In spite of the similarity in structure, the mechanism, the unknown mechanism, is different. And so here are just two of the bits of biological data. Note that this is the candida that we were looking at on the slide on invasive fungal infections, and that's the Aspergillus. And you can see that it has reasonable potency, not quite as potent as amphotericin, but reasonable potency. And more importantly, it's in vivo active, right? So this is an assay where you inject fungal spores into a mouse tail. You wait, and then you take out the kidney, and you see how many colony-forming units you isolate from the kidney. And what you can see, say for C. albicans, that at various doses you strongly suppress the viability of the fungus. It also is true, I don't have a slide to show it, but you can't kill mice with this, which actually is a virtue. So I don't want to make any great claims for this, but it is a path towards the discovery of a potential therapeutic agent following sort of a standard, if you will, natural product, chemical ecology kind of thing. And it raises a number of points that you can think about for the rest of the seminar. That is, how do you start finding things? Do you study nature, which is what many of us do, or do you study informatics, that is, large synthetic chemical libraries, genes, to figure out how you're going to do things? It also shows, I think, a big difference about how we look for things. We always look for a rapid, pronounced phenotype, like death. There's nothing like that. Whereas if you look for protective agents, you look for rather different phenotypes. We always look for a single active molecule. That's just the way sort of we were trained. We tend to then get rid of mixtures as we try to focus in on active molecules. And then when you begin a development pathway, you realize there's an interesting issue. If you're going in with a single agent, right, I mean, it's like not having a safety belt. You're going in without a backup when inevitable problems arise. So that's all I wanted to say. What I told you about today, everything was done in collaboration with these collaborators, Monica in her lab in Brazil, and Cameron in his lab at the University of Wisconsin, and Ethan and Tony did what I was talking about. So I thank them for the hard work. These people somewhat unwittingly funded it, and you once again for the invitation. So thanks.